Yeah, I'm going to start the live stream.
Okay, awesome. Yes, I'll just introduce you quickly before you start. We're very happy to have with us today Eleni Katifori. She started at Harvard doing her PhD with David Nelson, working on like very physicsy problems in soft matter, vortex pitting, and cleverly showing how they could explain the way pollen grains fold. She moved on to a postdoc at Rockefeller, I think around 2009, with Marcelo Magnasco. She also did work with Stan Leibler and lots of different, I might be getting a little bit of this wrong. I had to kind of piece together bits and pieces of information, but she did working on lots of different topics, applying concepts from soft matter topology and geometry to biological networks and plant leaves and phenotypic diversity in microbes. In 2012, she started a position at the Max Planck Institute, leading the physics of biological organization over there. In 2015, started at Penn and has been a professor there in the Department of Physics and Astronomy, working on problems at the intersection of soft matter and biophysics, exploring topology and biological networks, dynamical characteristics of biological materials. She's gotten many awards, the Simons Investigator, Burroughs Wellcome Fund, NSF Career, etc. And we are extremely excited to have her talk today on optimal trade-offs in biological flow networks. So thank you so much for joining us.
Thank you, Simon, for the invitation and for the very, very kind introduction. And you were mentioning stuff that I myself had forgotten, so it was quite impressive to hear such an accurate bio. So today we'll talk to you about something that like maybe colloquium style, something that we've been working on for a while, and maybe some of you have seen bits and pieces here, but I am really banking on the premise that I did a horrible job presenting the other time, so you don't remember anything and you get to say it again now. So my group is very, very much interested in vascular systems, and vascular systems are, I should say, in large-scale organism is inconceivable without a vascular or some sort of transport system. Even in things that are smaller than a single cell, typically they need some form of transport, and the reason for this is that diffusion is very, very slow. Right? Especially if you go to large-scale life, everything has a vascular system, and that vascular system is actually quite dense. In our own body, for example, the distance between the capillaries can be typically smaller than 40 microns. When we think about the vascular system, we mainly think about nutrient distribution, oxygen, right? But there's also many other roles that are played within the same system, right? One is waste removal, for example. Of course, in animals also have something like a lymph system as well for this purpose. There is also information transmission. So it's not only neurons that do this job, but also hormones. And the hormones travel through the bloodstream. So the same vascular system can play many, many roles. Now, there are very, very, very many flavors of transport systems in biology, and all kingdoms, let's say, all kingdoms that make large-scale organisms have it. Right here, and hopefully you can see my cursor. This is a picture, quite old picture actually, from the lymph system. This is veins. This here is Physarum polycephalum. This here is fungi. This here is picture of the vasculature of dicotyledonous leaf. And here, this cute animal is zebrafish, which is actually the model organism when people study animal vasculature. And the reason that it became the model organism is that it is transparent, at least in its early stages, and you can actually see very easily the vasculature. And you see it has a very, very well organized vasculature. Now, what you can see in all of these pictures, I mean, you can see a lot of similarities. You can also see a lot of differences, right? And let's say the emphasis of my talk today is to understand why living flow networks are so diverse. So one could say, well, they're optimal to different cost functions, right? Let's teleologically ask, what do they do? Well, they don't all care about the exact same thing. And in fact, within the same organism, they might care, the vascular system in different areas of the cortex might have different needs. Also, it's not only what you should build, it's not only optimality principles, it is also what you can build, developmental constraints, and also evolutionary happenstance, luck. Basically, you are the way you are just because of your evolutionary history. And of course, once one starts looking into these questions, the role of genetics will pop up, of course. And especially if we talk about humans, the importance for disease. And these two things, of course, in this talk, I won't talk about at all, but it is something that in general interests us a lot. Speaking of what interests us, this is a little bit very quick list of some vignettes of what my group has been doing in the past few years. Of course, I don't expect you to get anything from this slide. What I want you to tell you, though, is that the landscape of questions that have to do with vascular networks is immense. Because vascular networks are very versatile. We can have very simple models studying them, but they're also very, very rich, and they're also very important bells and whistles one has to think about that, for example, can have to do with the ability of the network to store fluid locally, as is the case, for example, in leaves or in flower petals, where local fluid storage plays a big role in how flowers bloom. There is also questions that have to do with resilience and what type of loopiness could make you more robust to disruption. There are more theoretical questions that have to do with topology characterization, right? Now, in this particular talk, I will be focusing on questions that have to do with maybe development, very briefly optimized nutrient delivery, and pulsatility. So in a very, very basic form, the workhorse I will be using to understand these networks is what you see in front of you right now, is basically a hierarchical networks of pipes, of tubes. Some are thicker than others. They are connected. There is current through a particular edge, which I will be mostly denoting with IJK, although the notation can vary a little bit. Each edge will have a conductance. You can think of an electrical network, for example. At each node, I have a pressure, and what drives the flow is a pressure drop between two nodes. And I have conservation of current at the nodes that you see here. And some nodes are going to be the so-called boundary nodes. This is what I will be meaning when I say boundary nodes. That means that the current is not exactly conserved, but there is a net input or output current. Or at those particular, quote, boundary nodes, I could be setting the pressure. These are the boundary conditions of the system. And there is a relation, constitutive relation, that connects the pressure drop with the conductance to the current. And if this was a linear Kirchhoff network, this would be Ohm's law. So at this point, I would like to remind you that you not only you're encouraged, but you need to interrupt me. I will be talking about two topics mostly, and I'm hoping I won't get to the second because you will be interrupting me so much that I won't have time to the second, but the talk is rather modular. We don't need to go over everything. So we talked about cost functions, and we talked about what these networks want to do. I see a hand up.
Hi, sorry,
Yes.
I had a quick question about the previous slide. So you're not assuming that you're not assuming continuity at any of the nodes, right? The sum of the currents at a particular junction can have a non-zero value.
Correct. But I said I know typically for most of the network, it will have a zero value. And there is going to be a subset of the nodes where I can set what that non-zero value is. And this is, you can imagine a closed system. You can imagine a closed system of pipes, like a water distribution network. And the boundary nodes would be the aqueduct that's feeding the system. There is one node over there, an input, and then the faucets at the houses are the also boundary nodes, the output of the network. The rest, that SJ is 0.
Okay, that makes sense. Thank you.
But I can also set the pressure on those boundary nodes, and this is also no problem. We can solve it. So I was talking about trade-offs. I was talking about cost functions, and in fact, what I will be talking about, it doesn't have to be per se exactly only on living networks. One could think of these questions and these principles for more generally for power distribution networks or for water distribution networks or for roads, but I won't discuss this. I mean, you can see them in the background here. So the cost functions can be very varied what these networks care about, but sometimes a network can care about two things. And these two things that the network cares about can be inconsistent. If the network really optimizes one of them, the other one will not be optimized at all. So the talk, what I will be talking about, will be focusing on this type of functional trade-offs. The first trade-off I will be talking about has to do with the fact that our vessels are compliant, that we don't have stiff arteries. So we'll be talking about pulsatility and the response dissipation trade-off, and we will see what that is. And time permitting, and I hope we won't have time for this, I will be talking about the built-in cost robustness trade-off, which in some ways it's easier to understand conceptually than the response dissipation trade-off. But it will also be, if we reach there, you'll see there are some kind of surprising aspects to it as well. So hopefully in this audience, we all have hearts, although nowadays this is not even certain. Now, the heart is needed to overcome viscous dissipation because the flow is viscous. Now, the flow, the vessels, I should say, in our body are elastic. And what happens is that when high pressure pulse comes in, the vessels expand. And when that pulse propagates through the network, it moves, the vessels contract again. And you can see this in this movie here, right? You can see the vessels expanding and contracting as the heart is beating. So a pressure pulse is propagating in the arterial system, right? Now, what is interesting is if we somehow could replace our heart, which is a pulsatile pump. I saw a sinusoidal version of it in this plot, with a steady flow pump of the same mean. It is easy to see from basic high school math that the steady flow pump will need less power to drive steady flow through the system than the pulsatile pump, steady flow of the same mean flow. Now, one could say if the body only cared to minimize the power needed by the pump, then the body should have perhaps evolutionarily developed mechanisms to kill that pulsatility altogether. So, okay, maybe we are constrained to have a pulsatile pump, but perhaps in most of the body, we convert that flow to steady flow, and we don't have to pay the price of pulsatility. And in fact, this happens, right? This is a cartoon here of pressure versus roughly, cartoon of the waveform as you go from the heart to the capillaries, and you see the amplitude of the pulse. This A, decreases as you go from the aorta to the small arteries to the arterioles, but it doesn't disappear completely even at the capillaries. And my question here is, why isn't pulsatility killed altogether so that we have maximum efficiency? It's not killed altogether in nature, and we'll see why. But it is killed. I mean, as people, we have made pumps, artificial pumps, to replace malfunctioning hearts that are called LVADs, left ventricular assist devices, that are continuous flow. It would be better if they had pulsatile flow, but they have continuous flow for engineering purposes. And so people they have these pumps as a bridge to transplantation because there are not enough hearts to do a transplant immediately. But these people cannot survive a very long time on these devices because they start getting some serious arteriovenous malformations, like things go wrong with their vascular system, and we don't understand exactly why. I mean, they can survive 2, 3, 5 years, but beyond that, people start getting serious problems. So let's say inspired by this, we started asking what happens if you reduce or you increase pulsatility or you remove pulsatility altogether in a flow network with elastic pipes. And to do that, we need to model, we need to have a simple model for flow through a complex network of pipes with elastic walls. And of course, we're physicists, we're not engineers, and we care about basic principles. So eventually, as I will tell you, we linearize everything, but anyhow. So we start from the basic nonlinear fluid mechanics, and then we assume rotational symmetry, and we assume the proper boundary conditions. We dimensionally reduce the systems, and we do all the proper approximations and simplifications for our case, which are basically the thin vessel approximations, moderate Reynolds number, and finally, as I said, we throw away all the nonlinear terms, and we make low flow velocity approximations. What this also means, because remember we have elastic walls, is that the area, the cross-sectional area, will increase linearly, essentially with the pressure. This effectively means that the vessel itself is like a capacitor. You store more fluid in the vessel will expand, and it will expand proportionally to the pressure. Anyhow, so I'm skipping many pages of linearizing and the Navier-Stokes and doing all of these dimensionality reductions, but the basic physics of the problem reduces to a mass conservation equation and a force balance equation. And in fact, the physics of the system can be reduced to something that most of you have seen before, and that's it's basically a transmission line, right? These equations, if I were to write them with circuit elements, it would be like a transmission line where you have the inductor here, which is the inertia of the fluid, a resistor, which is the viscous dissipation, and the capacitance, which is the fact that the vessel is elastic you put more fluid through it, you know, there will be more pressure. That's that relationship between fluid storage and pressure. And from this, from the resistance, inertia, and compliance, we can define some parameters like the length scale, lambda, and the time scale, tau. And then there's also an admittance because to match basically the units of flow and pressure. So we won't worry too much about it. But from the length scale and the time scale, we can get a characteristic velocity, and we can say, is this a number we should be worrying about with regards to how fast these pressure pulses that intrigued us right here travel? What do these numbers mean for the physics of the system? Now, as we will see later, not in too much detail, this lambda here.
Sorry, can I just interrupt?
Yes,
quickly?
Of course.
Can you go back to the last slide, the model?
Mm-hmm.
I guess I'm wondering, the area, the cross-sectional area is changing, right?
Yes. Yes. Mm-hmm.
So where's that?
Yes, where does this coming in? Excellent, thank you. So here there is a relation between the area and the pressure, right? And also that gives you a relation, of course, the area, it gives you a relation between the radius and the pressure. Here, this V is actually, this is bad notation, this is voltage, this should have been pressure.
Okay.
Right. So essentially several things happen when a pressure pulse is passing through. A pressure pulse is also an increased radius pulse. It will affect to different orders everything, including the stored fluid in the vessel, what is in the capacitor. It will also affect to higher order, but exactly what terms we should throw are in slides that I'm not showing you, but we can come back in the end. It also affects the resistance in a nonlinear fashion. So it affects many things. In the end of the day, if you non-linearize, you end up with this equation here, and it comes in the mass conservation. Just to I gave you a long answer, and I will shorten it. The most immediate place where it comes in is in the mass conservation equation, where here this CV, it should have been CP, because this V now is actually the pressure. If you go back to this equation, it's this CP, it's the cross-sectional area, and you see immediately if you have difference of the current coming in and the current coming out, you are storing volume.
Yes.
And this is where the cross-sectional area is coming in.
Okay.
Right.
Thank you.
It's in this capacitor, basically.
Right.
But you asked a very good question, which is kind of painful for us because it affects other things as well, which if you linearize, you lose some of it. But anyways, that's a tale for a different time. So this lambda here comes in. What this means basically intuitively is that there is a the amplitude of the pulse that the heart is sending out will be decreasing as e to the minus where you are in the vessel over lambda. And this is what the meaning of lambda, and we'll see the meaning of tau is basically, it is a proxy of how an individual pulse of specific, let's say, characteristic is propagating in the system. So if we vary, so we can play with these parameters here. We can vary the vessel compliance and fix all the other fluid properties, basically keep the network exactly as it is, and crank down or crank up how stiff or elastic the vessels are, basically give the organism atherosclerosis. And we can do this by changing lambda and fixing tau and the combination of alpha times lambda. Anyhow, this is a little bit too much information. So I see a raised hand.
Sorry, I probably missed this, but lambda would be determined by the viscosity, and
Yes.
I wasn't quite sure.
You missed it because I flew through it. So lambda is over here, is the characteristic length scale, which is related to the resistance per unit length, which is basically related to, it's like a Poiseuille flow, right? It's the viscosity, blah, blah, blah. The inertia per unit length, which is basically like the density of the fluid, and the compliance, which is how stiff or how elastic the vessel is. So it has all three.
Okay.
Of the, it's like the fluid and the solid mechanics together.
Okay.
So physically what happens is, to give you a feeling, is that let's say I send in, this is the heart, this is the vessel coming in, and then this is midway through the vessel, and then it will come back to this. And I send in a waveform, which is basically a step function. What will happen is that you will have a front, like a sigmoidal front, like this, propagating through the system with a velocity that is like lambda over tau. But how sharp that wavefront is will be the sharpness of that waveform is going to be broadening over time. And this is because the difference between phase velocity and group velocity. But let's see this a little bit, right? It's even a little bit more complicated than what I said. These are simulations. These are actually not even simulations. This is solving the equations analytically for a short vessel and a long vessel. On the left is short vessel. And what happens here, as you see when the simulation, maybe I'll walk through the simulation shortly. This is the sharp front I was telling you about. It's moving. But you see, before the system has chance to completely equilibrate, this front moves and reflects through the boundaries, right? So the system is dominated, for short pipes, the system is dominated by reflections. Here, something else happens. This is how essentially the vessel equilibrates, or the long vessel, the flow in the long vessel equilibrates to its new mean flow, which is 1. It goes from 0 over here to flow 1. And you see right now there are no reflections, but the vessel is kind of diffusively, the current kind of, quote, diffusively gets to its final position. So these reflections don't really matter. And the difference is the actual size of the vessel. Now, to make a very long story kind of shortest, we can calculate the time it takes for the flow in that single vessel to go from 0 to reach the new target value 1, because it doesn't happen instantaneously. It takes time for the midpoint of the vessel to reach the actual new value of the flow, to, as we say, respond to the flow. And this is the normalized midpoint current. You know, short vessels, it oscillates vigorously. Longer vessels, which is, let's say the red one is a longer vessel, you see smoother profile. Now, if I were to plot in log plot the residual current as a function of time, properly normalized, and we'll see what this means, all the curves basically fall roughly on the same line. And this line depends on a parameter here that we call a response scaling function. And this response scaling function tells us how fast essentially how fast the network is responding to the new boundary conditions. For short vessels, it's a constant, because you get dominated by reflections, and it doesn't change with length. But after a certain threshold, you get to a value here that is increasing with size, right? In fact, it's increasing This beta is increasing as the square of the length of the vessel. And this basically tells you, you double the size of the vessel, it takes 4 times as long to equilibrate to the new conditions. And here you can do it, this is the analytic prediction, just paper and pencil. This is the dust line, and here are simulations for different, essentially equilibrating to different waveforms, to different types of flow for different frequencies. But what is interesting is that you can also do complex networks, and these are cartoons. This is not the size of the networks that we did. And you get the same story. And all of these little dots are networks with very different architectures here. A different branching, random versus not random, loops versus no loops, all sorts of stuff. And you see, it's the same business. It's the same behavior for the response scaling function. And we don't have analytics for this. Somehow it works. Now, here, it was simple, what I put on this axis, the vessel size. But you have a network like this, and even more complex than this. What is the effective network size? What do I put on this axis? Well, there are coarse-grained parameters. That is a little bit like doing a path integral over the different paths between, I mean, this is your input, one side of the heart, like the arterial side of the heart of the systemic circulation. And this is the venous side in the heart of the systemic circulation. And here I'm basically taking different paths and I'm averaging the 1 over lambda of these paths, right? So, and there is also an equivalent, so there is the equivalent coarse grain parameter for the network. So this is for the single vessel. I'm using, to measure lengths, I'm using this normalized vessel length. And here I'm using this sigma, which is the network equivalent. And there is also a similar thing for a similar generalization for the pulse travel time. And it is, what is interesting here is that these results for the response time carry for complex networks with this some sense, this intuitive generalization. Although you can have multiple paths, you can have all sorts of stuff going on. But I promised you trade-offs. And here I've only told you about one, let's say, cost function, and that is how fast you respond. This response time is something that you need in the absence of active control, right? We know that we have the nervous system which supports our vascular system in the sense that if I stand up really quickly, you know, the pressure in my brain goes down really fast, but there is an active control of the vessels. They contract and they respond to increase that pressure. They contract to increase that pressure, and they respond faster than what you would expect from this, right? Which is great. So in a way, you could think of this cost function, which is the response time, how fast you respond to environmental changes, to changes of in your pump, as a lower threshold, which would tell you when you would need active control. Now there is the other part of the trade-off, which is the dissipation. And dissipation is actually very interesting. Now, as we said initially, and
Can I interrupt you one more time?
Yeah.
You just, I guess I'm a little confused as to how to think about this response scaling function. Could you
yes,
give a quick, like, physical?
Very quick. So not very quick, very specific instead of quick. So do you see this equation here?
Yes. Okay,
this equation basically is your residual current. Let's say you went for a run right now. You have your heart rate is 1 beat per second, right? And let's say you got up and you started running, and let's say this is not what happens actually exactly, but I'm oversimplifying things. Let's say suddenly, it doesn't happen that fast. Your heart rate doubled, something happened, right? Mm-hmm. Your capillaries would have no idea immediately that you started running. The muscle would be used. You would need more oxygen, but it would take, according to our calculations and experimental papers, references, it would take of the order of a few seconds, in principle, for your capillaries to know that. Now, how fast? Because the flow, it takes time for the flow to reach the new dynamic steady state. There are, depending on the complexity of your system, there are, fluctuations, but roughly your residual, the difference between the final and what you actually have, goes down as an exponentially fast, as minus, and the power of the exponent is minus t over tau. Remember, this is our scaling. This only has to do with the characteristics of the systems and nothing else, right? This is our naive scaling tau, our time scale, let's say.
Right.
Times a parameter beta, which corrects, let's say, this tau, a corrective parameter. Now, this corrective parameter beta is a response scaling function. It will tell you how fast you will equilibrate or how slow will equilibrate. If this beta is really large and in a long single vessel, it can be really painful, right? Because it goes like L squared. You double the size of the vessel, you double the length of the vessel, you quadruple this time, it will take you 4 times as long
to equilibrate.
Yeah.
Right.
So this is the response scaling function. It is this. The actual time is tau times beta, but tau is kind of trivial. You need the correction, and that beta is the correction.
Okay, awesome. Thank you very much.
And this number generalizes to complex networks. This was, I showed you for a single vessel, but the exact same things apply for a complex network. And we don't know exactly why, to be honest, but it does.
I guess now I'm more surprised about the flat part. Why is it ever a constant?
Because it is, if it is, because up to a certain, so you have two things going on. One is reflections. So because the equation in a way has a wave part and a diffusion part. And I'm not showing the equations here, but in the end, I can bring up the paper and I can show you. The final equation that will eventually, the terminal of all of these things has a diffusive part that kicks in at long wavelengths, and it has a wave part. And when you're dominated by the wave part, you're dominated by reflections. That is, cannot give you a better explanation than this, to be honest.
No, I think that helps. Okay, thank you.
So, and it's the same in network, it's the same in, but it's actually interesting though, because you see, we don't understand the point where it flips, the point where it becomes flat can change, and we don't understand. We get the scaling perfectly, but right. We don't get this part at all. So the other part that's kind of interesting is the fact that you also have the dissipation part, and this is where the trade-off comes in. So the stiffer you are, the more you respond very fast, of course, because stiffness here, and so I keep going, flashing the slides back and forth. Being stiff effectively means that the pulse doesn't decay or decays very slowly. And that means you will transmit the information that the pump changed conditions fast. So being stiff means that you respond fast, but at the same time, if you have a pulsatile pump and you maintain your wave form, you pay a lot in dissipation. And if you're very compliant, you're very unresponsive, but you don't pay a lot in dissipation. So in a way, there is a trade-off between the cost of fast information transmission and dissipation. And this is something you see in single vessels, you see in networks. It's a very robust phenomenon. Now, this is all in theory. What does it mean about the human body? And is it related at all to the human body? The biggest problem with the human body is that the flows are, you can't completely ignore the nonlinearities. However, you can argue that for the stuff that we're interested in, the nonlinearities are corrections. It turns out it's difficult to get a map of the entire human circulation, but these people actually did it, Minard and Smolitz. And they have a paper where they basically painstakingly reproduce the entire circulation, and they have they represent the microcirculation effectively. And actually that poses a few problems for us, but I won't discuss this exactly. And they also use nonlinear, they solve a nonlinear, computationally very expensive nonlinear system of equations to simulate the flow through the system. And then they compare it with situations where we know the waveform. So what we did is that we asked for their data and we compared their predictions with the predictions where we know the waveform and our predictions. And to make a long story short, although our model is linear, it's not that far off. So on this axis doesn't mean much. It's a very loose representation of how close you are to the heart on the arterial versus the venal side. We get the arterial amplitudes quite well. When we go to the venous side, we're more off, especially in the phase. But roughly for the amplitudes, we're okay. Actually, very okay, which is surprising, to be honest. So we can more or less trust our numbers. So what we do find is that indeed the our characteristic time scale, which is basically where we are on this axis, is about, the characteristic time scale is about 4 seconds. This seems to agree, and again, this is not published yet. This is preliminary. That's why you hear me not talking I'm not talking I'm not showing you any more about this, but it looks like based on when we are in the dissipation response function plot, is that the response time does matter. We're more on the high, fast response time side of the plot rather than the low dissipation part of the plot. We maintain pulsatility maybe for response time, maybe for mechanical integration reasons. Nobody knows, but we suspect it's for response time. But we maintain our compliance to the vascular compliance to values that promote a relatively fast response. So let's say the take-home message of this is that in the absence of any neuronal, which means active input, the network can adapt to changes in the boundary conditions, to changes in what the heart is doing, only with finite speed. It's not an instantaneous response. And this can be thought of, it's a limit on how fast the system can integrate and, quote, process mechanical information. The same thing is when I lift my arm, right, and I keep it up there, the whole pressure profile in my body changes. How fast will the flows, the average flow, will respond to this change right here, right? Or how fast will the rest of my body kind of know that I changed body position? Will adapt, I would say, to these new conditions. So the big question that we don't know at all, I'm just pausing it right here because it's really interesting, is what role could that speed have played in evolution? And also something that we're working on right now, but we have some very exciting preliminary data, but maybe in 1 year or two, what role this pulsatility plays in development. So this brings us back to these patients that have a non-pulsatile pump installed and they have these arteriovenous malformations. They lost their pulsatility and their vascular network remodeled. So we're trying to understand theoretically why this happened. So I could stop right now, or I could talk very quickly about building cost and robustness trade-off if you want. I can let you decide. What would you like?
I think we can keep going for 10 minutes and then switch to a discussion.
Perfect. So I will show you now this, which what you see here is a network. And in fact, it's essentially the placenta of an egg, of a fertilized egg. It's the corallantonic membrane. And what you see here is the network grows, but it also massively remodels and acquires hierarchy. This is what happens to any embryonic development. In fact, this is very well known, right? This is very well known since very old times, right? Initially, when there is no heart beating, there is recruitment of cells essentially, and you make some sort of very primitive, disorganized vascular structures like the ones shown here. But as soon as you have flow, you get hierarchy. And there's a lot of work phenomenologically establishing that the reason is that the vessel, there is mechanosensing, the vessels measure the wall shear stress and they adapt. Now that fact was actually known from observation for a very long time. And it's been popularized, this equation that describes it, or versions of this equation that has been describing, has been popularized recently, relatively recently in Physarum, but it has been known for much longer than this paper. So it's valid for Physarum, slime mold for arteries and veins. It's not exactly this equation, but it's a sister equation to this that has to do with the shear stress, but it's all flavors of this equation. And if you add an extra term, you can also get the development of leaf vasculature, but the terms here mean something different. So this is in a way the equation that I'm showing you here with or without this constant production term, the G, the equation that tells you that the rate at which you increase your radius or your conductance is a positive function of the flow. You use the vessel a lot, it will grow. If you don't use it, there will be this term here which stabilizes it. So that vessel will shrink or disappear. So this equation is use it or lose it equation here. It's very general and it's been used for many things. So what we did, and very briefly, is that we took this equation with this constant production term. And if you look at what this constant production term is doing, says in the absence of flow, at steady state, in the absence of flow, you will have a disorganized network. So basically the problem that it solves is that the use it or lose it equation without the constant production term will say if you don't have a heart, you have nothing. Whereas the use it or losing equation with a constant production term tells you if you don't have a heart, you have a disorganized uniform network. So we take this equation and we solve Kirchhoff's rules, essentially, but we apply growth boundary conditions. So as a function of time, more and more blood comes through the system because the chick embryo is growing. And then we solve it. And to make a long story short, what we find is that you start with nothing, then you develop this network. So in this case, the boundary conditions, the blood comes in here and gets distributed evenly in the whole domain. And then this becomes hierarchical. And what you can play with the timing of growth on this axis and how important this constant production term is. And you can see that your final product, the developed network, will be an optimum of dissipation in the end. And if you're building the network while growing, which is you're here, that means that this is the importance of the growth term essentially, and this is the importance of the time scale of growth, right? If you're right here, you will have found the global optimum. And when you're right here, you will have found only a local optimum. If you build something where you're not growing, it's going to be suboptimal. Now, the salient points of this, very briefly, is that very simple developmental equation here. If it runs with growth versus no growth, it can give you an optimum for dissipation. So it will give you something good for dissipation. But if you are growing while you're building, it will give you a very good optimum for dissipation, right? And the story could have ended here. This is an equation that optimizes dissipation, and it optimizes dissipation really well. It actually uses only local information, which is great, right? If you look at this equation, each vessel only knows its current and its conductance. It makes a decision based on that conductance. But in the end, the final product will be a global optimum of dissipation. But there's more than that. The flows are dynamic. So I have an extra degree of freedom. That means that as the system is growing, or as the system exists, maybe the flow wants to go from here to these red dots, maybe it wants to go there, maybe it wants to go there. So I have an ensemble of flows. Now, unlike what I was showing earlier about pulsatility, the system immediately equilibrates to the new current values. There is no pulses here. It's in a way, it's a primitive way to do fluctuations, but there are fluctuations. There is some sort of dynamics. And depending on how strong the fluctuations are, this simple equation will give you different phenotypes. And what I can do is that I can take those phenotypes and go back to my trade-offs and just measure what these networks are good for. And let's say there were two things. There were two objectives. One was cost, how expensive it is to build it. And the other one is percolation penalty, which is basically how fragile you are. So right here, the gray one are buildable things. If you're down here, that means you can build something which is very cheap and very robust. There's no way you can do that, but you will be right here, right? And this line, of course, if you're right here, that means you can move somewhere within the realm of possibilities, the gray possibilities, and get something which is better in both objectives until you reach this front, which is in economics terms, it's called the Pareto front, which is the front of optimal compromise. You cannot improve one objective without hurting the other. And this is a bad biological example that I will skip. Now we can do the same thing for this developmental equation. We can talk about fragility and cost, for example. Actually, we talk about three things: cost, fragility, and dissipation, efficiency in a way. We can make all of these networks, and then we can populate this in our case, three-dimensional space, we can find the Pareto front. Not everything is in the Pareto front. This is not, for example, this is not, but these two are. And then we can put them in a line. And what we have is that we have essentially a spectrum of phenotypes. And that shows us a trade-off. So on one side, we have low-cost networks, but are not robust, and they're very hierarchical. And on the other side, we have high-cost networks that are robust and uniform. Now, this is all produced by the single developmental equation. So you see there is this trade-off between these two quantities, the robustness and efficiency, in a way. So our entire vascular system for the long-range transport arteries and veins to the capillaries can be described qualitatively with the same equation. Now, this is kind of old work now, 2019, and I'm skipping this. But we've now, we've looked at more recently with my student, Yorgos Gounaris, we've looked at what happens when we take we add something extra to the cost function. We consider dissipation, we consider cost, but we also consider distributing nutrients. So now I have an extra field that I have to worry about, and that is oxygen. And to make a long story short, again, I now have a trade-off. And the trade-off now is how uniformly I perfuse my domain, or how efficiently, with regards to dissipation, I transfer my fluid from one end to the other. If this was an organ and I prioritized perfusion, I would get something that looks like this, depending on if I have low metabolism or high metabolism, right? If I prioritize dissipation, I would get something which would not perfuse quite well. Here, yellow means you don't get a lot of food, whereas orange means you get a lot of food, right? But of course, it is possible to try to be good at both, to compromise. And in that case, you have something like this, right? This is a trade-off. But depending on what you prioritize, you can either equiperfuse, or you can basically be efficient in your transport. So I think here I will finish. I don't want to take any more of your time, but on this talk, we try to talk about optimality to different cost functions and various trade-offs that we want to face. What we did not talk about, though, at all, or almost at all, is the effects of nonlinearities. I mean, we have valves, and where are they in these landscapes? And also, we talked about this a little bit, but I haven't shown you any results. What is the role in general of fluid dynamics in evolution and development? This is a version of my group. Now, we managed to take a more recent picture than this, but I haven't put it in yet to update this monstrosity here. So some people have left, some people are still here. The people that did most of the work that I have shown you were Sean Fancer, who did the pulsatility work, Henrik Ronellenfist, who did the adaptation work, and Yorgos Gounaris, who did the perfusion work, and also Tatiana. I didn't show you any of her slides, but she also did some very interesting work related to the adaptation and perfusion. Okay, so with this, I would like to thank you very much for the invitation and for your attention, and now we can finally discuss.
Thank you so much. I think why don't we start off with questions about the talk? So I'll start it off. So in the second portion of your talk, describing these generative equations for vascular networks, is it the case that a set of generative equations can essentially create the diversity of vascular networks that you see across the tree from fungi, lower organisms, to vasculatures in higher organisms?
That is my hope. That is, I would like that to be true. Of course, if you talk to biologists, they will tell you that the situation is much more complex than that. But sorry, I'm trying to move my slides. Okay, there we go. So there we go. So this is it, right? So here, without this G, this equation is exactly what people speculate already for Physarum. If you take the equations that people use in papers about shear stress, mechanosensing, blah, 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 for arteries and veins, and you change the parameters, you end up to something like this, to the functional equivalent of, it's not exactly the same powers here, but the steady state is in the same class of this equation, the same class as this. So bells and whistles are different, the core is the same. And if you add this constant production term, you get the oxygen canalization model for the leaf. Now, about fungi, I don't know. The biggest difference, and now I have to go to this slide. The biggest difference in my feeling comes from, where is it? Show you something. If you look at fungal networks, for example, you see they're not the tracks are not necessarily, just by visually inspecting them, right? They're not necessarily so hierarchical as slime molds, right? And you can ask yourself why. Perhaps there is a developmental constraint that tells you, no, you cannot, you cannot build tracks, you cannot build vessels above a certain width. Here, for example, this network is not hierarchical at all. Now, why is that, right? In principle, the equations that I was showing, you could produce it. But also, this is the lymph network, it has valves. There are no valves in the equation I showed you, right? So, to some degree, you can argue that this and this and this have the same equations, are governed by the same fundamental equations, but there are situations where the bells and whistles are so strong that that equation is not enough.
I see. And one follow-up question now that we're on this picture. Is it the case that the genetics of vasculature is strongly conserved, or does it sort of arise independently many times?
I would say, from what I can tell, I'm not a geneticist, but from what I can tell, independently many times. This is why it's so impressive. I have not included this picture in this talk, but in situations where, for example, you have to build a planar network with kind of similar constraints, geometrical and functional constraints as a leaf, as is in some cases in the chorioallantoic membrane, for example, in the egg, where we have a lot of data. It is, by eye, it's kind of difficult to distinguish. It is amazing that they look topologically so similar. And these are organisms that became multicellular independently, right? This is the genetics is that different.
That's incredible. Avi?
I just had a clarification question. Could you just remind me, maybe I missed it early in the talk, what the biological value of dissipation is, like why you think the network would optimize for dissipation? Oh,
this is a great question. I had a number here. Did I read it? Sorry. Yeah. So I've calculated it myself, then I forgot the answer, to be honest, and then I found this, I think, in a paper. So you can actually calculate back of the envelope how much energy calorically the heart takes to pump the blood. And it is actually, it's not like the brain, right? The brain is extremely power hungry, but it's not something insignificant. So it is to the organism's advantage to have a flow system that requires little energy, and that dissipation equals basically the energy that the heart, that the power that the heart needs to run the system. So it's not as dominant as the brain, but it's not insignificant. Even worse, if you need a lot of energy to circulate the flow, you need a better muscle, you need a bigger muscle, which can create other problems. So there is at least some evolutionary pressure to build an efficient network because of the pump. Does this answer the question?
I think so. I have to think about it, but I
Good.
appreciate the answer.
And I see another hand.
Yeah.
Manuel, yes.
Thank you. First of all, great talk. Thank you very much. I was just curious. I couldn't stop thinking about like scaling the system to, I don't know, a blue whale. If I now suddenly I'm chased by some orcas and my heart starts pumping much faster, and I need a fast response. Like, what does the blue whale have to build differently? Like, is it more stiff veins or arteries? And do we know actually, like, have people measured that kind of thing so that to compare it with what your model will predict?
Oh yeah, this is a great question. The very short answer is that, and I will almost quote it from a paper, there is almost no literature in comparative vascular physiology. This is terra incognita. We know humans very well. We know some basic models organism very well, but the questions that you're asking is really not answered. There is some stuff about general, how the metabolism scales with size in all of this big body of work for allometric scaling, which is not exactly what you're asking. It's kind of related how the metabolism scales with size and how fast, for this reason, the heart is beating with size. But that's not your question, and there is no serious data for your question at all. It's very interesting, though. We are now trying to do something which is maybe along those lines a little bit, but it is very primitive. So I can't tell you more. Okay, thanks. And the second question that I had was that you mentioned that maybe we have pulsatile flow, maybe to respond faster, but is there any example in evolution, I don't know, like a crab, or I don't even know how they circulate things around their bodies, but some other organisms that don't use pulsatile? Because I cannot imagine how would you have like a continuous flow. Oh, another great question. So there are two ways that biology has figured out how to push the flow. One is by, in a way, peristalsis. By peristalsis, meaning, and this is our heart evolved from a very primitive peristaltic system, right? Basically by having a tube and periodically squeezing it, right? So there is muscle around the tube and the tube gets squeezed and you push the flow like this, right? There is Imagine this is the tube.
Yeah.
So this is how circulation started, by having muscle around this tube and doing it like that. And for reasons that actually completely different project, I'm not going to talk about it. When you do that, you need a valve. And depending on, well, when you do that and it's low amplitude, you need a valve. And then valves started evolving. And from that, you started having a heart. And our heart, which is a 4-chamber heart, is only one design. There are other animals with different numbered chambered hearts. You don't have to have 4 chambers, right? So it's a whole, very, very interesting. So that was one mode. The second mode of transport, which we also have in our body in some places, it is very, very subdominant though, is ciliate flow. So the vessels themselves inside, they're coated with cilia that keep beating, and the flow is pushed because of the cilia. I don't know a lot about it though. I know it's important for primitive organisms like jellyfish. This is how they circulate their flow. And I think that is all in all, from what, and I know very little about it, is more of a continuous flow. Everything else is really starts as a pulsatile flow because of the peristaltic pumping in a way. Other complications that make systems in different animals very different is open versus closed circulatory systems. Like we have a closed circulatory system, but the jellyfish, for example, has an open circulatory system. And many other animals that you mentioned, I think the lobster has like a hybrid in a way. So it is anything you can imagine, nature has built it, to be honest. But most of it, when you have any sort of peristalsis or peristalsis derived, it is pulsatile.
I see. Okay. And in plants, it's not active.
Oh, yeah, I forgot the plants. Yes, this is all animals. Plants, it's not active, it's entirely passive. So you don't care that much about dissipation in plants. But that's I can talk until tomorrow, right, about plants, but I won't. So plants is different.
All right, thank you very much. Really interesting.
Eleni, if we can have you for another